to introduce Dr. Paul Duquesne from Northwestern, associate professor, really one of the leaders in electrophysiology of the channels. And he is going to tell us today about pathogenic variants in the poor region and how that relates to channel dysfunction. For the participants here, what I'm going to ask is that at the end of the lecture, you raise your hand and unmute yourself. You'll be called on, unmute yourself, and then ask your questions directly, because I think it fosters a better communication between the speaker and the whole group. So with this, Paul, I'm going to turn it over to you, and thank you again for taking the time to meet with us.
You bet. You bet. Thanks a lot. Thanks a lot, Brad. And thanks, Terry, for inviting me out to give a talk to you guys. Happy New Year. I haven't been able to attend any of these this year, but maybe that will change going forward in 2025. Maybe that's my new Year's resolution. Okay. I'm frozen. There we go. All right, great. So I won't belabor the point because you all here are experts in ADPKD, common monogenetic disorder. Skipping forward to the biochemistry, where the variants associated with the disease affect either PKD1 or PKD2. We know from the biochemistry that they can form homomeric channels, as well as the functional data from native and overexpressed systems. PKD2 is a member of the polycystin subfamily of TRPs. They localize to the primary cilia and ER organelles. They seem to escape electrophysiology characterization from the plasma membrane. Our lab has been making a lot of effort in characterizing variants associated with PKD2 that are associated with the disease by fabricating, small electrodes, as Brad was kind of implying, and recording these single channel recordings, which are carried by the homomeric channels. So these ones are constitutively active, voltage gated, but calcium modulated. We know a great deal from regarding the biophysics of these channels, whereas the PKD1, PKD2 complex is much more of a challenge to characterize. Thus far, it seems to be forming a channel, or a channel-like receptor, where it associates with a 1 to 3 stoichiometry, and forming perhaps a ligand or autoproteolytically cleaved ion channel in cilia or other membranes. So I'm going to primarily focus on the effects of polycystin 2 variants and within the context of the homomeric channels, because they are most tractable using our techniques. So despite our strong understanding of the genetic components involved, PKD1 and PKD2, this monogenic disorder, we don't really know how the variants impact how they cause disruption of the channel itself. Our understanding is pretty limited. So as initially characterized by Hao at University of Utah, the PKD2 homomer forms a domain-swapped ion channel architecture. It has pore-forming regions here in green, a top domain, which is analogous to the ML external domain, and a voltage sensing domain. And this is a true voltage sensing domain because it has a couple of gating charges, which are among the most highly conserved gating charges in amongst voltage gated channels. So these channels, when they're domain swapped, they form this tetrameric architecture, and the disease-causing variants are found all over the channel, but they primarily aggregate in this top domain. And previously, we made an effort to characterize, in collaboration with Hao, the mutations found within the finger 1 region of this part of the channel. to characterize its loss of function. But for today's talk, I'm going to be primarily focusing on three variants which are found in the which are associated with ADPKD and are found within pore helix 1. So that is phenylalanine 629S, cysteine 632R, and R639C. And so obviously you can have missense variants or perhaps nonsense variants that cause ADPKD. But what we're going to see now that while types of mutations matter, you can have a missense variant which causes really drastic changes in the both the biosynthesis of the channel as well as the and trafficking as well as function. And despite their close proximity, you can have very different impacts. So not only is it makes sense to characterize what the variants do, we don't know much about the mechanism controlling the state dependence, driving the conformational changes in these channels, shifting them from the closed to the open state. It's really poorly understood. So we a few years ago carried out a full screen in the polycystin 2L1 channel, which is a close homologue of PKD2 or called PKD2L1. And the missense variant, we screened the entire pore region of this channel. And what we found is that many alanine substitutions in the external part of the channel actually drove gating defects in our functional analysis. And so with that, as well as with a number of different conformational states that have been found with different trip channels, where the external side of the channel is found in a kind of a dilated state, there's been a proposed hypothesis about how these channels open and close or gate. And one competing theory is that while everyone accepts that the internal gate is involved in opening and closing the channel, others have also proposed that the external side of the channel also dilates or gates. And so by interrogating those sites in the pore helix, we can also examine, okay, do we actually see gating defects? And if we take it a step further and structure it, do those gating defects cause conformational changes in the ion conducting pathway? So first, we start off with just HEK cells and use overexpressed the missense variants in the pore helix with an HA tag. And so these cells also overexpress an ARL reporter such that they can visualize the primary cilia while the cells are still alive. And what we found is that the length of the cilium actually gets smaller when the missense variants are overexpressed. And I should add that this cell line does not have either PKD1 or PKD2, so that we are really studying these channels in isolation without contributions of endogenous polycystins to these human embryonic kidney cells. And so when we actually look at what happens with the channel and how does it traffic using 3D sim, so we're able to visualize the primary cilia in 3 dimensions such that we can subtract out any kind of underlying fluorescence so that we can be sure what we see in that 3-dimensional organelle is actually from the signal from the channel itself. What we see is that we have 2 variants that pretty much traffic reasonably well to the primary cilia, but we have this cysteine substitution to R in the pore helix that doesn't traffic at all. And so with this in mind, we already see that the missense variants, even though they're within just, a few dozen residues of each other, localized in the same motif of the channel, different disease-causing variants could have very drastic different effects on the localization of the channel in primary cilia. So these cysteine mutation is essentially a complete loss of function in the primary cilia itself. So we carried it forward into doing some cilia electrophysiology. So in this case, what we're doing is we're just applying steps from minus 60 millivolts and stepping to a depolarizing state, depolarizing membrane potential. And what you see is you have open channel events that occur throughout these traces. And over that duration, we can assess the total effect on open probability. So what we find for the missense variants, the ones that do traffic to the primary cilia, we have big shifts in the voltage dependence of opening these channels. So we have shifts in the voltage of the half maximum potential between 27 to 42 millivolts, which is close to 2 to 2.5 kilcal mole in energy, change in free energy. So given that the total amount of gating that is required to open, or energy required to open this channel, is somewhere on the order of 1.5, we've effectively doubled the amount of energy it requires to open these channels. So clearly this is a loss of function, and a significant one at that. But it dwarfs, or in my opinion, it's totally relatively less high impact compared to the cysteine 632R variant, which doesn't function and doesn't traffic at all to the primary cilia. So in this case, we've patched over 20 primary cilia. We don't see a single channel event. So with this in mind, we have clearly a story that's shaping up where we have 3 different variants that we've characterized functionally in close proximity to each other. They're just simple missense variants. What is the molecular mechanisms that are responsible for these divergent effects? So in order to investigate that further, we started to do cryo-EM in my laboratory. So over 3 years ago, I attended several of the national centers, their workshops. And so, after attending that, I applied for in-residence training. So in this case, I was able to go out to Stanford for 2.5 weeks and just learn cryo-EM. And so you can do all this with purified protein in hand. And the reason why I'm telling you this is because I want to encourage, maybe for the new year, a lot of the young residents or young PhDs or students to apply for these. It can be done. And the best part of all is it's free. So all you have to do is come up with the time and some money perhaps for lodging. This is actually my first shot from an overnight data collection with polycystin 2. So I was able to do that once I had reasonable biochemistry. So it's, again, I'm not saying it's easy, but I'm just saying that, it can be done if you're inspired to do so. With that, so that was about a little over 3 years ago. So with that, I wanted to build the infrastructure here at Northwestern, and I was finally given funding from a strategic investment from our dean's office, as well as applied for and granted some infrastructure block grant through the state of Illinois. We have purchased and now have in-house our first cryo-EM facility that we call FACET for Feinberg Advanced Cryo-EM and Tomography. So we have now capabilities of these instruments. So this is a 200 kV cryo-EM microscope. And we have acquired a FibSim for milling for tomography purposes. So in this case, you could use it to look at molecular interactions in situ, so we're avoiding now the artifacts that are associated with overexpression. And we're working towards and trying to identify donors, of course, for Krios Titan. But nonetheless, we have a Krios Titan that has just gone in up at our Evanston campus, so about 30 minutes away. So I've been named the director of this facility, and we are also taking external those who want to pay to use the microscope at our facility, and go ahead and reach out to me if you're interested. I don't think that we'll be able to provide training within 2005, but I would encourage you to start the National Center battery if you're interested in getting started, and then perhaps circle back for additional training here at Northwestern. So we saw that one of the variants didn't localize to the primary cilia. Why is that? If we look at some of the basic biochemistry of this channel through size exclusion, we can see that the arginine 638C variant still forms a tetramer along with the phenylalanine substitution, but the cysteine 632R is actually falling apart and is producing actually a bit more monomers and less of the tetramers or the oligomeric structure or oligomeric protein. When we use glow melt to examine the stability of these channels, we can see the blue trace where that cysteine substitution actually never hardly fluoresces, and that it indicates that over this temperature range, or really within this whole temperature range, that this protein is just not stable at all. It's never going to form oligomers so that we can't structure it. Whereas the other variants more or less are slightly perturbed, but we can still get usable particles. So we resolved these structures. They're about 2.7. This some of the higher resolution structures to date. You can see that some of the resolution falls off as you start to move to these externally flanked parts of the protein. But keep in mind the area that we're really interested in, the pore, is sitting well around 2, between 2 and 3. So we're pretty confident in our structures as far as our maps go. We can see that the channels that we can structure actually still form their domain swapped architecture, and when we overlay it with the wild type channel, we don't really see any big changes in folding, suggesting that these missense mutations aren't really driving massive differences in the overall assembly of these channels. So we have one set that retains its native native architecture overall, the 629S and the 638C, and then we have one that we have no chance at structuring and doesn't localize to the primary cilia. For the ones that do go to the cilia, still form channels. What is the molecular cause of that, of their gating defects that we observe in our cilia electrophysiology? So if we look locally, we can see that the substitution of the phenylalanine 629S, this side chain really adopts the same conformation, but it is sitting within a hydrophobic pocket, which is a little bit unstable, not very descriptive. Then we also look at the arginine mutation, which is participating in a tripartite hydrogen bond network in the just outside of the selectivity filter. We see that cysteine substitution just breaks that disulfide bond. Again, both structures locally still are preserved, and the external gate more or less looks unperturbed. However, if we start to look further down the ion conducting pathway, what we observe is that this hydrophobic gate, the length of this restricted length, actually extends in the mutations, which are more or less occurring up here. So we look at this other side if we looked at the 639C mutation, we can see that it becomes almost completely asymmetric. So with this all this change is primarily driven by the change in the orientation of just a few residues. So one of them is this N681 carboxamides, which becomes optimized at hydrogen bond distances between 3 to 1.8 angstrom. We can see it's a bit more snug in the variant channels. And in one case, it's actually completely asymmetric. So these interactions really actually illustrate a really fun observation, which kind of turns the one gate or two gate hypothesis on its head, meaning that if you have what we have is a long range allosteric effect, where if you have mutations that affect the packing within this pore helix, it will transfer to changes in the lower gate, such that the functional effects that were observed previously in PKD201, which indicated that the external site might be a gate in and of itself, actually aren't. What is being described is truly an allosteric effect of just one gate being impacted, because otherwise we would have seen it within our structures, our interpretation anyway. So using super resolution and cilia electrophysiology, cryo-EM approaches, we discovered mechanistic differences on with a group of 3 mutations in the pore helix. So these effects are really ranging, affecting either channel function or the channel's ability to assemble and form channels in the primary cilia. The resolved cryo-EM structures have ranging effects on the collapse of the gate for the ones that do function, while the other ones just completely keep the channel from forming their oligomers. And these unexpected findings have significant implications when you think about rational drug design. So, on one hand, you might approach a variant by generating a channel modulator, something that will affect the functionality of the channel. On the other hand, you may want to get something that binds and rescues the folding. So something like the corrector approach of Vertex. But you could see that perhaps by characterizing all these mutations or different mutations within the channel, that some things are not going to be so simple, as far as if you have a missense mutation in the pore helix, you're going to have a folding problem or gating problem. We can see that it's actually a mixed bag. And so perhaps this opens up an interesting approach for either, or at least maybe we should investigate new, including both channel modulator type drugs, as well as those that are correctors in themselves. And so this was a fun study also, just from the examination of the biophysical determinants of gating of polycystins, and that we see that, at least in my opinion, I believe this supports a one-gate hypothesis where mutations in the external side of the channel are primarily driving allosteric effects in that lower gate. This is my joke. Happy New Year. Oh, it's so awkward when you can't hear people laugh. Okay. What are some New Year's resolutions or future directions, right? So we would like to establish AD-PKD mouse models based on PKD2 variants. And I think you should stay tuned for what Patricia Oteda is going to publish this year. And I would also like to address the elephant in the room. And the elephant in the room is PKD1 structurally and defined variants, and how they impact the PKD1-2 structure. We're moving towards the era of outside of overexpressed systems and starting to move more towards tomography. We're hoping to build upon that. And of course, get drug-bound complexes, both the homomeric and heteromeric form. And I think that by doing so, we can perhaps establish a blueprint for tomorrow's therapeutics. I want to thank the people that actually did the work. So primarily Ori Asarte Palermo. So he's been my postdoc now for 3 years, and he's been with me since the outset. He is originally an organic chemist and a really good one at that, and he transferred his organic chemistry skills to the world of biology and mastered cryo-EM. He is on his sixth variant so far, and he continues to do outstanding work. And I suggest keep an eye on him. I think he's a rising star. Eduardo Guadarrama, who's our a graduate student in the laboratory. He's a neurobiologist, but I have persuaded him into examine polysystins, including the polysystins that are found in the brain. And he is actually graduating this year. So he's another rising star. I want to thank the NIH and for the funding, and thank all of you for hearing me out today. And of course, the PKD Research Consortium, Resource Consortium. Thank you very much.
Thank you, Paul. I'm going to open this up for questions. I'm going to start with one. Based on that structural changes you see in the pore, do you think there would be a way as a treatment, just thinking down the road, of engineering another variant that could be co-expressed with that variant that would change the structure back?
Wouldn't that be fantastic?
Yeah, but would it
It'll be
work?
fun. I think worth a shot. So perhaps you can dope in maybe through some kind of gene therapy of overexpressing a gain of function to perhaps override the loss of function that
Yeah, so what I was wondering is you chose those changes in the bonds, right? So could you engineer another variant to be expressed with that one that would then shift it back to a more symmetric?
yeah, well, or symmetric or just a different or just a more open channel. I think a good candidate would be a Yang Yu's 604P mutation that's non-disease causing, and put that in. Yeah.
And the other question I had was there's always this debate about haploinsufficiency or loss of heterozygosity type of thing.
Mm-hmm.
If you express a wild type along with some of these variants, does it rescue the channel opportunity? So if you get a mixed channel with a mutant and a wild type, what's the impact of that? You may not have done any of those type of studies.
I haven't done those studies, but I can see how it could work. The trick would be, how do you convince yourself you've expressed, how do you convince yourself you've expressed both proteins at the, equal proportions or sufficient proportions? You're going to have, of course, in the background wild type, because they will just form with the amount that you produce there. They will form channels on their own. But yeah.
All right, questions? Please raise your hand and unmute yourself. And looks like I'm occupying all the the questions here. This is unusually quiet.
I can ask the question because I don't know how to raise my hand. So here is my question. Ludmila Sabataro from Hopkins. I know we looked into the PC2 and the PC2 binds to CFTR. When you measure the channel activity of all these mutations, did you by chance look at the chloride activity? Question one. And now second question would be, if you would use the CFTR correctors, knowing that they bind to the CFTR, I know in fact because we did the experiment, CFTR binds to PC2, goes to the cilia together, in the CF knockout mouse, the cilia is affected. Would you actually consider to use the CFTR correctors to see actually if you can increase the function of this channel?
Yeah, I think that's a really good approach. I think it would just be fun to actually see a full battery CTFR correctors, both one looking at rescuing cilia trafficking, just on and of themselves. As far as but I have not, for example, looked at any of the CTFR correctors or the prototypic vertex polycystin corrector, as far as looking at how they affect the function of these variant channels. I think that's more to come. As far as the question as it relates to chloride conductances, no, we have not yet isolated any kind of chloride conductance from the primary cilia. I'm not saying they're not there, but if we change our recording conditions in the form of the types of anionic charge carriers, perhaps we can unmask a conductance if indeed these polycystins and CTFR are interacting, and perhaps in the cilia or otherwise. So yes, on both counts, we still need to look. I think that's a great question.
Leaf.
Thanks very much. Great talk. Really exciting and very much outside of my area of expertise. So I'll ask a very like naive question. That when I look at the cryo-EM data and the structure data that you present, like when you talk about moving forward to more tomography of endogenous structures, could you expand a bit more about what the limitations there are and how, like, you know, for example, would it be possible to image these channels on cells from an organoid, in an organoid, you know, or what kind of limitations you're seeing?
100%. I think the limitations is feedback that is now very prevalent in the cryo-EM community in general, which is we need to move away from overexpressed systems, because in a lot of cases we are there. You could leave it open for an artifact because you are overexpressing that protein, purifying it and only it, and then structuring it. So it leaves open what are its endogenous regulators. So those things that, in one fantasy of mine, and something that I've been talking a lot with Ivana Ko about, is examining what are the ultrastructures in both primary cilia and ER membranes, and what do they mean? Like, how do the structures differ? And why that is great is because you get to see what the structure, what all the components are that are natively there in both organelles. And by doing that, so you're, getting closer to what is truly endogenous. And the way it works is that the approach involves isolating, whether you're using kidney cells or others, but milling layers of the sample with a focused ion beam, and do more or less doing lithography in the form of cryo-EM to reconstruct total complexes in their native environment. And so what I'm saying, I guess, reason why I want to move into that for 2025 is because I want to get closer to what is natively happening in the cilia or other places. So I think that we're probably in for surprises as far as the types of things that are associating in both organelles, as well as it will give us a new glimpse on how we can perhaps stabilize these proteins for new therapeutics, and as well as just understand the basic biology of the dysregulation that is occurring in response to disease-causing variants. It's a good question. It's an area I'm really excited about, but
thanks. It's just one simple follow-up question is, like, from my naive perspective, when you take a 293 cell and wipe out its PKD2 and then re-express it, you get tons and tons of these proteins at the surface. Is it going to be crowded with other things that you want to ignore if you're looking at endogenous cells or endogenous expression?
Oh, right. Yeah. If you want to look at endogenous expression, it would be cool actually to bring the expression a little bit more under control instead of just like overexpressing it with the virus. But I think that you can perhaps put tags in frame with perhaps a kidney cell, whether it be from a cystic cell that you isolate from a donor, or perhaps a cell that has been donated, or a wild type like human donor cell from the collecting duct. And that tag, you can perhaps put a sun tag in vivo to actually visualize where the cilia, where the channels are, and then carry out your milling. So this is an area called light correlated electron microscopy. And so what it is not only are you having an EM laser involved, but you're actually involving now an additional confocal laser. And this way, you can actually visualize under endogenous expression your protein of interest as it's expressed in kidney cells. That's the dream.
Fun. At least I think it's fun.
Oh, yes. Thank you. Yeah. Hey, great talk, Paul. Here's Wen Qian. Yeah. I have a question, has something related to what Brad was asking. So I'm thinking about a ADPKD patient with that, say, with the one of point mutation C632R, where you show in your overexpression system that the protein was not stable and do not traffic to the cilium, right? But this is basically this is the homozygous situation where every single protein you express contain that mutation. But in a patient, we know heterozygotes, where you say in most of the tubule where they're normal. So let's assume that those cells, they're not cystic or early stage, and you will have half of them are wild type allele, the other half containing that point mutation. Suppose the expression is 1-to-1, so you're going to have half of your protein subunit is wild type, the other one is containing the mutation. So, but since they're normal, if they don't have second hit, how do you think about it? Then those tubule must have a hybrid or molecule. Presumably they are defective in gating or something bad, but they're still normal. How do you approach that?
I'm not quite sure. So as I understand it, my understanding of the disease is that, and from our analysis of PKD2 models expressing PKD two variants, that the second hit needs to occur in order for the disease to progress. And so if part of like what you're saying is the critique, like, oh, you're overexpressing a variant and you therefore have a homozygous variant in these HEK cells, no argument there. Most certainly that's a limitation to the study, but it also helps us get at exactly what the defect is likely going to be if it's going to propagate in its association either with other PKD2 homomers or perhaps a variant or even a wild type PKD1. So I think the fun thing to do at this point is the field probably shouldn't, let's just start structuring every single combination of a variant, because it's just going to go on way too long.
Right. No, but Paul, I think I meant a little bit different. I meant in heterozygotes, where they're normal, they're not like heterozygous mouse, PKD2 mouse.
Yeah, but that mouse is not cystogenic, is it?
Right. But half of the channel had the heterotetramer contain that mutation, right? So you would think that in a normal looking tubule with the heterozygous mutation in it, and would I not expect to have a chimeric type of channel where you must expect somehow defect, structural defect, gating defect, but yet they're not cystic.
Yeah, but I don't think you can rule out that the channels, the homomers, the ones that are from the native functioning polysystem 2, PKD2, whether or not those are still forming from the wild type, because he says heteromer, right? You can't rule out
Yeah.
that limited amount of functional channel is not sufficient to keep off disease, right? To keep to stall disease progression. I think it's an interesting question, and if I understand it right, is also like, is it possible that some of the missense variants can be kind of poisonous, right? To the tetramer, right? Like, so the missense gets involved, and so there's lots of hypotheses that could argue either way. Like, one example I could think of is whether or not gating itself is cooperative, in that you need all 4 subunits to open in order for the channel to function, and one poisonous subunit is sufficient to drive loss of function or complete loss of function for the entire channel. I think
you could test that in your system.
Yeah. I could, but I think it will probably it probably will have a bit of a mixed bag as far as the caveats. Like, you'll probably end up with 2 pools of channel. I mean, 2 pools of data, and it might be hard to parse the 2. It would be a dream to, for example, generate a concatemer where you have 4 copies of the same channel, and then you just put the mutation in one subunit at a time and see how long it takes, or how many mutated subunits it would be required to drive the loss of the channel function. It'd be a great way to kind of test the cooperative gating hypothesis. Right. Yeah, well, could happen. Yeah. I think I wanted to, what time is it?
It's 3:45. We got some time yet.
Let's talk about the elephant in the room. Is that okay?
I think we have a whole bunch of questions right now yet.
Oh, okay.
So I see Dr. Barr, Maureen. We can come to the elephant in a second.
Paul, you did make me laugh when you said you couldn't hear anybody laugh, so I did laugh. I have a cell biology question.
Sure.
So for your variants that do traffic to cilia and cause shorter cilia, what are your thoughts on that?
Yeah, that surprised me, because so we initially went about in our 2020 paper, I think it was 2020, but like, we overexpressed the variants in HEK cells in the background of a PKD2 null, and we actually saw no difference in the finger 1 variants in the length of the cilia. And so I was expecting the same thing here. However, I neglected to remind myself that these were done in a PKD1 and PKD2 loss of expression channels, cell line, so that now that what we see here is likely, or undoubtedly likely, to the extra loss of PKD1 in the background of these channels, which really unmasks its cilia phenotype in these cells. Excuse me. So one thing we did notice is that there is not like a complete loss of the cilia itself. So does that mean that budding cilia or cilia formation is calcium dependent? It would, maybe if it's coming from the cell body, it's hard to know what is the cause of it. We'd probably have to go downstream of it. Perhaps there are calcium dependent expression of different cilia proteins that are being impacted by the loss of the polycystin expression. Yeah, downstream of it. That's my thoughts. Sorry, all I got is genetics and structure function. Sorry, Maureen.
Ying.
Nice talk. Well, nice discussion, particularly because I came in late and I only heard you saying like PKD2R1. But I didn't hear the rest of it. So when did you talk about the PKD2R1? What was new there and so on?
Yeah. So the reason why I mentioned PKD2L1 was because I was setting up the hypothesis of the 1 versus 2 gate hypothesis. So initially we took 2L1 and we mutated every single residue within the pore region. So this is data that we published a few years ago. And what we found is that you can get gating defects in the external side of the channel. And so, as well as on the inside of the gate. So that suggests that, okay, maybe there is perhaps two gates. And so one of the side quests of this manuscript was to explore a little bit of that. And actually what we found is contrary to it, in my opinion. That is, we don't see big changes in dilations of the local environment where the mutations are taking place. Rather, we saw allosteric effects on the internal gate. And so for me, it seems like it's more leaning towards a refutation of the two-gate hypothesis and leaning more towards the one-gate hypothesis with allosteric regulation through the external side of the channel.
Ah, okay, great. Thank you so much for explaining it.
Anthony.
Okay. This is mainly a structural question, since you've got such nice resolution of overexpressed PC2. Often when you do Westerns of overexpressed PC2, you see polymers of PC2. And I'm wondering if you see any evidence of isopeptide bonds in your cryo-EM reconstructions.
Isopeptide bonds.
Right. Like non
Forgive me, I don't even know what it means.
Okay, non-disulfide bonds, bonds between residues that have like OH groups and amino groups.
I see. I don't think that we have seen anything that it's running very differently when we do Western blots of our overexpressed system, like suggesting, sorry, when you see an isopeptide bond, you see almost like a smear, or what would it
No,
look like? Because I imagine that, go ahead.
Just discrete bands at the multimers, at 2, 3, 4 subunits linked together, but in the presence of either DTT or beta-mercaptoethanol.
With the presence of the reducing agent, you see it.
With the presence of the reducing agents, you still see oligomers.
I'm not sure. I don't think I have looked at several Westerns I haven't seen that, but that's probably due to a condition that we have. This will be fun. If you don't mind, email me later. I want to know why that is.
Sure.
All right, Paul, I'm going to turn it back to you to address the elephant in the room now. I was going to ask specifically what happens when you put this in with peak 81, but
yeah,
maybe
so
where you're going.
yeah. So I think it kind of dovetails with a lot of this good discussion we've had. So we're getting to the point now where we need to start using a little bit more AI to help us out, because we can do structure after structure of various variants in combination with or not, but that's going to be a tremendous undertaking, especially when you consider the kind of heterozygosity or transheterozygosity that you typically find in the disease. So one of the things that we're wondering about is like, okay, so what did the disease causing variants do to the 1-2 complex, the kind of elephant in the room? And so I think if a good approach, and one that we've recently undertaking, is using a lot more AlphaFold 3. And so we have in our hands, and they've been available now. Our coordinates, we released them 4 months ago. So they've been available to the community to download through PDBs. You can take those coordinates now and ask AlphaFold to assemble complexes with PKD1. And I understand this is very hypothetical, but this is the year that David Baker was awarded his Nobel, and I think that it would be silly not to start examining some of this, at least for hypothesis generation, right? And so one of the things that we've done is the PKD1 structure is resolved by Jia Ganshi's group is powerful and tremendous and came way ahead of its time. But there's a lot of limitations, and I think that they would agree with that. And there's big swaths of the channel, well older than 70, almost 70% of the channel not structured within that. And some of the things are even within the pore itself. And so that's the exact hotspot that we need to start to examine. We're going to look at what the variants are doing. So we asked AlphaFold to render it. You know, how would you fold it? We generated well over 6 models with PKD1 based off the coordinates of our missense variants, and see how it assembled. Everything looked pretty similar to what the wild type is, but AlphaFold now can complete sections of the protein that were otherwise dark, including locations within the pore. So let's see if I can get this to work. So there is not a helix here, and obviously if we ask AlphaFold to render it correctly or render what it expects it to be, we can see that there's a nice interface being formed by this model. And within this model, we can see that, for example, there's an arginine hydrogen bond interaction with a with a serine from PKD1. So we can clearly see that this would disrupt this missense mutation located here would disrupt normally what is an interaction between the heteromer. And so I think this is just an example of like, okay, at some point, we're going to have to stop and actually maybe use some of these AI tools to help us understand what are the more complex structures with PKD1. By no means does that replace cryo-EM, but I think it's an important tool that I think everyone should be dipping into by now. One of the other missense variant, the phelanine 629, is also the same as you would find in the homomeric forms from the cryo-EM, and that it just more or less occupies what is normally a hydrophobic pocket, not particularly informative. But if you ask AlphaFold, I thought this was fun, because really these are kind of fun tools. You never know what's going to come out the other end. When you challenge AlphaFold to fold the variant structure, the one that actually doesn't fold itself in cells, it kind of there's like a screw up in the code. So this arginine, when it's substituted for the cysteine, when AlphaFold folds it, it creates bonds that can't possibly exist. And I think that this kind of tells you, one, the limitations of AlphaFold. Two, it also tells you that it struggles with what a cell would struggle with assembling this, in that this is a very tightly packed space, and this creates way too much steric class to ever fold properly. And so it kind of gives us a little bit of insight as about what's hard for the GPUs is also hard for the cell. Yeah. And I think that much of our loss of function that we see in much of the loss of function that we see with the homomers is most likely going to transfer in a similar loss of function way to what you see with the heteromers.
I've always been curious about the AlphaFold 3 and comparing what that generates versus what the actual cryo-EM shows, and particularly with your variants, if you were to model those, does the AlphaFold 3 agree with what the cryo data says?
Reasonably. So it's just kind of, there's never one answer, right? You input your sequences, and depending on how much bandwidth you have, you can generate a suite of models, and from there you'll see there's some that seem a little bit surprisingly different, but primarily they look pretty close to what we get by cryo-EM. However, none of them predicted the allostery that we observed by cryo-EM. So it's yet again another example of why you need to do the actual experiment and actually do this arduous wet lab stuff to come out with more accurate predictions.
We're going to have jobs for the next 15 or 20 years until AI takes over everything.
Right.
So Paul, thank you. We're just about out of time, but thank you for a wonderful presentation and excellent discussion. I just want to bring up one resource that we have developed in the PKD ORC that, I don't know if this would be useful to you, but the We do have a PKD2 halo tagged cell line, mouse, and all that. Those renoepithelial immortalized, temperature sensitive immortalized epithelial cells are available. If they are of use to you, please reach out to us, and we're happy to share that resource with you to hopefully push this forward.
Beautiful. I think that's a great idea.
So that way, and it's the cells are marked with SST or with or without SSTR3, so you can see the cilia and live cells, and it's the endogenous.
This is perfect for cryo ET.
So let me know, and we're happy to share that resource with you. Okay.
Excellent.
All right, Paul, thank you very much for an outstanding talk, and thank you for the audience for great discussion.
Thanks a lot.
Yeah.
See y'all. Have a good new year.
Yeah, you too.